Hello, my name is Max Porter, and my research deals with the effects of lead nitrates on Planarian regeneration. Now, this research will evaluate the effects of heavy metal exposure on the vitality of stem cell-like cells. Lead is known to be a very adverse pollutant in the environment. It is widely known to be a precursor to many birth defects. A few of these defects include developmental delays, neurological damage, and slowed postnatal neurobehavioral development. The development of embryos is largely due to stem cells. These cells are proliferant, meaning that they are able to develop into any cell type needed to be found in the cell of an organism. These stem cells regulate the growth and development of a fetus. Lead exposure is known to cause developmental delays and slow growth of a developing fetus. There is a possibility that exposure to lead causes an acute effect on the stem cells found in developing embryos. Planaria are a freshwater flatworm which have the ability to regenerate lost or amputated tissues. As seen here, you can see that an amputated planaria with its head cut off can regenerate its head after a period of roughly 2 weeks to a fully grown planaria. Now, Planarian regeneration is due to an undifferentiated cell population found throughout the planarium called neoblasts. These neoblasts are proliferant cells and very similar to stem cells. When the planaria have lost tissues, these neoblasts migrate to the affected area and begin regenerating the tissues that were lost. And the regeneration of neoblasts is very clever in the fact that if the head of a planaria is amputated, it is capable of regenerating the entire body of the planaria. Even from a specimen or a piece of the planaria that is roughly 250 cells, it is capable of regenerating the entire planarian body back. Now also, if the planaria is cut bilaterally down the down the center of the planaria, as like that, then the planaria, or the neoblasts actually, are able to regenerate the planaria in full of a completely complementary half of the planaria completely back. Now, Planarian have been tested with heavy metals such as cadmium, nickel, and copper. Previous research has shown that an increase in the concentration of heavy metal exposure results in longer regeneration times. Longer regeneration times signifies that the regeneration is being inhibited, while a shorter regeneration time would show that it is being influenced in a positive way. Now, the heavy metal exposure also shows many behavioral changes, and Planaria have been concluded to be a good bioindicator for evaluating the effects of environmental changes, such as heavy metals contaminating their environment. Now, my research will be composed of applying several concentrations of lead nitrates to the planaria. I will then amputate the heads off of the planaria and measure the time it takes for the planaria to regenerate. My data would then be collected and analyzed to see if there's any significance in the actual effect of lead nitrates on Planarian regeneration. Thank you, and yep.